Welcome, everyone, to our journey into the fundamentals of biomolecules. Today, we are going to explore one of the most elegant and crucial mechanisms in all of biology, base pairing. If you have ever wondered how a tiny microscopic cell knows exactly how to copy its genetic blueprint without making catastrophic mistakes, you are in the right place. We are going to build our understanding from the ground up, so even if you are completely new to this, you will soon see the beautiful logic hidden inside our DNA. Let us begin by picturing something from our everyday lives. Imagine a jacket with a broken zipper. If the teeth on one side do not match the teeth on the other, no matter how hard you pull, that jacket will never close properly. In the microscopic world of our cells, DNA acts very much like a perfectly designed molecular zipper. The two long strands of DNA are held together by tiny chemical teeth that only fit with one specific partner. This perfect, exclusive matching system is what we call base pairing. It is the ultimate security system, ensuring that the two strands lock together tightly and accurately, protecting the precious genetic information hidden inside. Now that we understand the zipper analogy, let us look at what those chemical teeth actually are. In biochemical terms, base pairing refers to the specific hydrogen bonding between nitrogenous bases on opposite strands of nucleic acids. There are two main types of these bases. First, we have the larger purines, which include adenine and guanine. Second, we have the smaller pyrimidines, which include cytosine, thymine, and uracil. The fundamental rule here is that a large purine must always pair with a small pyrimidine. Why? Because this specific combination ensures that the overall width of the DNA double helix remains perfectly constant from top to bottom, preventing any structural bulging or pinching. Knowing that a purine pairs with a pyrimidine leads us to the specific partnerships, often called Watson-Crick base pairing. It is not just any random pairing. Adenine has a strict, exclusive relationship with thymine, and they hold hands using exactly two hydrogen bonds. Meanwhile, guanine always pairs with cytosine, and they form a slightly stronger connection using exactly three hydrogen bonds. This strict complementarity is incredibly powerful. It means that if you know the sequence of bases on one strand, you automatically and completely know the sequence on the opposite strand. They are perfect mirror reflections of each other's chemical needs. Before we move on to the mathematics, let us see this with a concrete visual example. Look closely at this schematic diagram. On the left side, you can see the larger adenine molecule reaching across to bond with the smaller thymine molecule. Notice the two distinct dashed lines between them. Those are the two hydrogen bonds we just discussed. Now look at the right side. Here we have guanine pairing with cytosine, and you can clearly count three dashed lines connecting them, representing their three hydrogen bonds. The vertical structures on the far edges represent the sugar phosphate backbone, which acts as the sturdy fabric of our molecular zipper, holding all these teeth in place. Seeing these bonds visually brings us to an important question. Why does this strict matching system matter so much to living organisms? Simply put, without base pairing, life as we know it would not exist. When a cell divides, it must give an exact copy of its DNA to the new cell. It does this by unzipping the DNA and using each single strand as a template. Because adenine only pairs with thymine and guanine only with cytosine, the cell can perfectly reconstruct the missing half without making mistakes. Furthermore, scientists exploit this exact mechanism in modern medicine and forensics, like in PCR testing for viruses or DNA fingerprinting at crime scenes. This perfect biological copying mechanism naturally creates a mathematical pattern, which brings us to the work of a scientist named Erwin Chargaff. Long before the physical structure of DNA was fully understood, Chargaff analyzed the chemical makeup of DNA and discovered something remarkable. He noticed that the quantity of adenine in any sample is always equal to the quantity of thymine. Similarly, the quantity of guanine is always equal to the quantity of cytosine. Because of this, the total amount of purines will always perfectly match the total amount of pyrimidines. This fundamental biological law applies to all double-stranded DNA across all living species, from bacteria to humans. Now that we understand Chargaff's discovery conceptually, let us look at how we express it mathematically. The formula you see here states that the sum of A and G divided by the sum of T and C equals exactly 1. Let us break down these symbols. The brackets around the letters represent the molar concentration, or percentage, of that specific base. The numerator, A plus G, represents the total amount of purines. The denominator, T plus C, represents the total amount of pyrimidines. Physically, this means if the percentage of any single base increases, its specific partner must also increase by the exact same amount, keeping the overall ratio perfectly balanced at 1. Now that we have our formula, let us apply it to a classic problem. We are given a sample of double-stranded DNA where 20% of the bases are adenine, and we need to find the exact percentages of the remaining 3 bases. First, because adenine pairs exclusively with thymine, thymine must also be exactly 20%. Next, we add them together, telling us that A and T account for 40% of the entire molecule. To find what is left, we subtract that 40% from the total 100%, leaving us with 60% for guanine and cytosine combined. Finally, since they must be present in equal amounts, we divide 60 by 2. Our final answer is 20% thymine, 30% guanine, and 30% cytosine. Physically, this shows a perfectly balanced genome. Having successfully calculated base percentages, let us shift our focus to the physical strength of the DNA molecule itself. The stability of a DNA segment heavily depends on the total number of hydrogen bonds holding it together. Remember, every AT pair contributes 2 bonds, while every GC pair contributes 3 bonds. This means that a DNA segment with a high concentration of GC pairs is physically stronger and more stable than one with mostly AT pairs. In a laboratory setting, scientists need to know this because it dictates exactly how much heat energy is required to melt or separate the 2 DNA strands for experiments. To quantify that physical strength, we use a specific equation to count the bonds. Here is the formula for the total hydrogen bonds, denoted as H total. Let us look at the variables. N subscript AT represents the total number of adenine-thymine base pairs in our segment. We multiply this number by 2 because they share 2 bonds. N subscript GC represents the total number of guanine-cytosine base pairs, which we multiply by 3 because they share 3 bonds. Adding these 2 products together gives us the grand total. Physically, if the NGC variable increases, the H total increases rapidly, meaning the DNA strands are locked together much more tightly. Let us put this bond counting formula into practice with a concrete example. Imagine a short DNA segment that is exactly 50 base pairs long, and we are told it contains exactly 15 adenine bases. We need to find the total hydrogen bonds. First, since there are 15 adenines, there must be exactly 15 AT pairs. So, NAT is 15. Second, we subtract those 15 pairs from the total 50 to find that there are 35 GC pairs remaining. Now we plug these into our formula. We multiply 15 by 2 to get 30 bonds from the AT pairs. Then we multiply 35 by 3 to get 105 bonds from the GC pairs. Adding 30 and 105 gives our final answer of 135 total hydrogen bonds. This tells us the segment is quite stable and heavily reinforced. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that base pairing is the ultimate biological matching system, holding DNA together. Second, the rules are strict. Adenine always pairs with thymine, using 2 bonds, and guanine always pairs with cytosine, using 3 bonds. Third, this relationship naturally leads to Chargaff's rules, ensuring the ratio of purines to pyrimidines is always perfectly balanced. Finally, this simple but elegant complementarity is what ensures your genetic information can be safely stored and accurately copied. You now have the master key to understanding how genetic mechanisms function. Great job today.